So amyloid is one mechanism, but it's not the only mechanism. What are some other mechanisms, Mark, that should be explored, or there are some data that that there may be hope that they may be efficacious that are going into phase 2 or phase 3?
There are dozens of other studies that have been going on, and thankfully we do have a relatively large pipeline of studies, particularly for Alzheimer's disease. One area is looked at secretase inhibitors, trying to inhibit the enzyme that leads to the formation of the toxic form of beta amyloid in the first place. There have been a number of different studies looking at that. To date, efficacy has not been proven, and so it's unclear the future of those particular studies. A number of different experimental agents looking at different ways to offer some neuroprotection. So even if we are unable to stop the buildup of amyloid and tau, is there a way that we can protect the cells? Is there a way we can reduce inflammation, which appears to cause so much of the damage? And so there's a number of different agents looked at that. And there's been some strategies actually using agents that are often used for diabetes, but to try to boost the metabolism of brain cells and see if that can have some impact as well on the course of the disease. So lots of ongoing issues. I think we know this is a complex disease, and it's really slow going. The failures of some of the major studies have been disheartening, but lots of different mechanisms being explored, and whether it's one or a combination of these, hopefully something will yield some really robust findings in the near future.
That's good. I'll bring it back to you, Brad. Frontotemporal disorders oftentimes involve different forms of tau. And of course, there's a different kind of tau involved with Alzheimer's disease. What are some approaches that are being looked at for Alzheimer's disease and tau?
Yeah, the portfolio is growing of compounds and strategies to target pathologic forms of tau protein, which is nice to see, because whether it's Alzheimer's or whether it's the primary tauopathies, we really need that tool in our toolkit. So monoclonal antibodies, and the initial thought was, how can you use a monoclonal antibody if the problem with tau is inside the cell, intracellular? But it turns out that this is where this hypothesis about the transsynaptic spread of tau comes in, because if tau is actually pathologic, hyperphosphorylated tau is released from the synapse and crosses to the next cell, and that may be the basis of its spread, there's a chance to intercept it with a monoclonal antibody. And so that strategy is being tested. There are a variety of other small molecule strategies that are being tested to try to see if either you can reduce the process of hyperphosphorylation, potentially through modulating enzyme function, or potentially find other ways to clear out the pathologically hyperphosphorylated forms of tau so that they don't keep spreading and depositing as tangles, or the forms that we see in the frontotemporal disorders as well.
So it sounds like all these, now that we're developing our armamentarium of biomarkers, are going to be very, very useful to be able to actually measure those processes and have them be sort of targeted.